member from our sister university, I can say, because Davangere University was earlier Kuampu University PG Center. We have with us today, as a part of our last session, Dr. Santosh Kumar M. So, Dr. Santosh Kumar M is a student of Gulbarga University, where he completed his MSc Biochemistry in Gulbarga University in 2006. And later he completed his PhD also from the same department of biochemistry from Gulbarga University in 2013. So his previous positions include as a senior analyst, he played a role of senior analyst in TV SUD South Asia Bangalore, and from 2014 to 18, he was a scientist at ROSM division of Aurobindo Pharma at Hyderabad. So later he joined Davangere University as assistant professor in Department of Biochemistry. So he has 13 publications to his credit, and his his impact factor, it ranges up to 6.5. So that's a testimony that how quality research he does, and how good papers he has produced. And when coming to his contribution to the field is concerned, he is a expert in biotransformation as well as drug metabolism. So he actually developed a plate method for screening of bacteria capable of degrading aliphatic nitriles. Then also he contributed for a plate method for, he contributed for genome of a novel isolate of Paracoccus denitrificans capable of degrading the N-dimethyl formamide. So then he also contributed to the purification and characterization of amidase from Paracoccus species, utilization of amidase inhibited whole cell for bioconversion of acetonitrile into acrylamide. So from his bio, you might have now figured that he is more of a expert in biotransformation. So today he is going to tell us something about microbial enzymes and their application in pharmaceutical industries. So I know him from a couple of years since his joining to our department, because Department of Biochemistry holds a special place in my heart. So so he's a very knowledgeable man, and he has a very good experience both in corporate and academia. So with this brief introduction remarks, I welcome Dr. Santosh Kumar M to this session, and I request him to kindly take the session and give us some scientific deliberations. So please share your screen and please start, sir.
Thank you, Dr. Santosh sir, for introducing me to your webinar. And once again, I thanks Department of Biotechnology, Kuampu University, Kuampu, for considering me as a part of this national webinar series, Decovidizing Biotechnology Research. Thank you, sir. Thank you for inviting me and giving opportunity to discuss with enzymes, applications of enzymes in pharmaceutical industry. So before going to start my topic, I want to introduce myself. So I started my career for isolation of novel microbial culture for industrial use purpose only. So I started basic research, but this basic research have novel opportunity, novel approaches to use this basic research into a applicable research. So I started the organisms isolation, characterization, and use of these organisms, microbes for the treatment of toxic chemicals. So I completely involved in biodegradation of organic solvents, that too nitrile degrading, nitrogen containing organic solvents like dimethyl formamide, acetonitrile, acrylonitrile. This because these nitriles are commonly present in the environmental effluent, because these nitriles are versatile organic solvents, and amides are also organic solvents for more use in pharmaceutical industry for extraction purpose and other process purpose. After extraction and everything, they will discharge into the environment. So nowadays, the environment become more polluted because of polluted pharmaceutical waste also, and contribution of other waste from different industry like agricultural, plastic, pesticide synthesis industry, pharmaceutical industry, drug industry, and different acrylic sheet industry. So these nitrile organisms which are present in the environment, they are capable to use these toxic nitriles and amide solvent as a sole source of carbon and nitrogen. Using these toxic chemicals, microbes are growing and they are surviving in using as a carbon and nitrogen. So for that, I isolated the nitrile degrading and dimethyl formamide degrading organisms using as a sole source of carbon and nitrogen. Then we developed a technology, immobilized technology for treatment of dimethyl formamide, which is present in the industrial effluent, lupin pharmaceutical industrial effluent. They are using dimethyl formamide for extraction of rapamycin. We successfully demonstrated the use of immobilized whole cell bead for treatment of dimethyl formamide. So then I developed a indicator plate method for screening of nitrile. So these nitriles are cyanide substituted organic solvents. So it is commonly, these nitriles are commonly used in synthesis of acrylic sheets and acrylamides and acrylic derivatives in pharmaceutical industry. So these nitriles are very toxic because this contains a cyanide group, organic cyanides. Nitriles are organic cyanides. So this C triple bond N is not easily break by simple method, but the living organisms having potential, they contains nitrile hydratase, nitralase, and amidase plays a very important role to break this C triple bond N. So I used this microbe for development of indicator plates, simple indicator plate to develop screening of toxic nitrile or detection of nitriles in the environmental samples. So then I moved to application of this enzyme. Not only we can not only use these organisms for the treatment purpose, we can also use these organisms for the application purpose. So because these nitrile hydratase, amidase, and nitralase enzymes have potential application in synthesis of many uh, pharmaceutically active drug molecules, intermediate or precursor molecule synthesis. So then I concentrated on applications of microbial enzymes for potential useful product synthesis, useful product production. So, as you know, all the enzymes are specialized class of proteins, have specialized class of proteins that act as a biocatalysis. Because of existence of this catalytic activity, many chemical reactions like anabolic reactions or catabolic reactions, whatever the reactions takes place in the living system, are because of enzymes only. So these enzymes are important biomolecules in the living system. So using the catalytic activity or catalytic property of this bio enzymes, we can exploit this catalytic activity into a synthetic form or production form. So as you know, enzymes have many applications. Enzymes can be used as a biocatalyst. Enzymes can be used in R&D centers, research for research purpose, like development of recombinant clones using the recombinant enzymes as a tool for generation of recombinant clones. We can also use enzymes in diagnostic purpose and we are also using enzyme in a beverages and food industry and poultry and poultry industries. And we can also using enzymes in pharmaceutical industry as a therapeutic agents. So nowadays we are looking for the enzymes for the biofuel productions, the energy productions, and household items. So these enzymes have many applications. So this enzymes, application of enzymes, are use of microorganisms for the production of useful chemicals or useful organic solvents or acids. So this is not a new field. The approach is new. The application of enzymes or application of fermentation technology is not a new field. So it is already exist and well established. The use of suitable biocatalyst for the useful or important product productions, that is very important. So uh, so so enzyme become a big market in pharmaceutical industry. So how is big enzyme market, and which part of the countries are accountable for large enzyme market shares, and who are the key players in enzyme market, and what are the factors driving enzyme markets? So this the enzymes global market size is estimated to be 9.9 US dollars billions in 2019 and is expected to reach 10.6 US dollars billion US dollars in 2020. And the most of the largest market or largest part involved in enzyme production or use of enzyme are making enzymes as a useful product in North America dominated the enzyme market with a shares of 37.4% in 2019. This is accountable to increased demand of why this is because they are using enzymes in pharmaceutical industries, biotechnology sectors in US market. They are looking for the green technology, greener manner, production of biopharmaceutical chemicals using green technology. And they are advanced in enzyme technologies. And who are the key players in the market? What are the companies involved in production of enzymes and use of enzymes or supply of enzymes? Some of the key players includes enzyme Novozymes, DuPont, ESN, amino enzymes, Lonza, Codaxis, Merck, and in India, Codaxis, Merck, applied enzymes, advanced enzymes, Arozymes, Novozymes playing a very important key role in market. So what are the factors driving the enzyme market? The key factor that driving the market growth including increasing demand of enzymes in industries, especially in the food beverages industry and pharmaceutical industry and animal feed sectors. So this pharmaceutical industry is becoming new area use of enzymes. So it is occupying the space in the market of enzyme market. So if you, the global market estimation shows that in 2019 and expected in 2020 is $14.7 billion, increasing in demand. So in India, the enzyme production, according to Industrial Enzyme Market Forecast Opportunities in 2020, Indian industrial enzyme market is estimated to exceed 361 million by 2020 on account of increasing pharmaceutical industries, application of these enzymes in pharmaceutical industries, and application of enzymes in food processing industries, and use of many enzymes, especially in the textile manufacturing facilities in India. So India and China are the leading in Asia specific region production use of enzymes. So enzymes have many applications. So in pharmaceutical applications, so enzymes become a socialitic need. So we are using everywhere enzyme knowingly or unknowingly. For example, in pharmaceutical industry, different therapeutic proteins are used. Different enzymes are used for the synthesis of drug molecules and development of diagnostic kits or developments of tools for the detections. Enzymes are used in beverage and food industry. Already it is existed, it is present, and all the food processing enzymes are used in food processing and other purposes. So even we are using some of the enzymes in detergents and Nowadays, more developing countries are concentrating on biofuel industries using enzyme for the production of biofuels. So apart from this, so natural gas conversion like methanol conversion and fine chemical productions are more recently considering the use of enzymes in industries, especially for fine chemical productions includes enantiomers, stereoisomers, and specific neurotransmitters. So because of selectivity, regioselectivity and specificity, so enzyme will become more important, especially in fine chemical productions. So nowadays becoming everywhere we can apply the enzymes. So I am concentrating on only on the enzymes which are what are the enzymes used in food industry, detergent, biofuel, and other industries. So I am concentrating on applications of enzymes in pharmaceutical industries, especially in pharmaceutical enzymes. Different enzymes are used for the different purposes like nutraceuticals enzymes. Some of the enzymes used in the nutraceutical for the purpose of supplementation of nutrition along with lactose, invertases, lactoses, invertases. Some of the enzymes involved in uses as a therapeutic agent for the treatment of diseases like collagenases, asparaginases, tyrosine, ureases, and some other enzymes. So in the diagnostic purpose, some of the oxidoreductases, glucose dehydrogenases, alcohol dehydrogenases, and aspartate aminotransferase, alanine aminotransferase. These enzymes are used in diagnostic purposes. As enzymes also used as a therapeutic agent like proteins, proteases, trypsins. These are used as a therapeutic agents. In digestive aids also, digestive product development also, enzymes have applications for the preparation of amylase enzymes, pectinase enzymes, lipase enzymes, sarcosidase enzymes for digestive syrups for treatment of metabolic disorders. So some apart from these, the pharma industry also looking for the enzymes for the synthesis of drug molecules using the pro molecules are intermediate for synthesis of active pharmaceutical industries. Why this drug synthesis become more important using enzymes? Because the enzymatic reactions are highly specific and the product contamination is very less, and the pharmaceutical industry becoming the pharmaceutical product becoming day by day more complex. So synthesis of more complex by using chemical method and enzymatical route method, we can synthesize new xenobiotic molecules or new pharmaceutical complex molecules. So this can be achieved to produce stable biocatalytic solutions, stable. So these pharmaceutical industries we can achieve by using enzymes to produce scalable biocatalysis catalytic solutions used in the pharmaceutical chemicals, fine industries by reducing the process, by reducing the raw material cost, although it is beneficial to green technology. In that way, we are producing using biotechnology, using the biotransformation method, that is a green tech, it is a solution for green technology. It is a solution for remove toxic chemicals, or it is a solution for less contamination or reducing the contamination in the environment. The most significant advantage of using enzyme as a catalyst is because highly regioselective, chemoselective, and selective enzymes, which use only particular product, either L form or D form or S form or R form. So selectivity are more important use of these enzymes. Another important thing is the high degree of product specificity. The large required pharmaceutical processes due to streamlining the product synthesis routes or subsequent improvement of the process in economics. For example, the synthesis of complex pharmaceutical drug molecules require number of steps. So we can reduce the steps by using high degree product specificity enzymes. We can by By reducing the steps, we can synthesize complex molecules like neurotransmitters. These neurotransmitters are already present in the living organisms. We can use enzymes for the synthesis of intermediate for synthesis of neurotransmitter drugs. The use of appropriate enzymes not only removes the stereoselectivity and specificity, it also enhances the yield of required or desired stereoisomer. For example, if it is a racemic, so we need a one enantiomer or one stereoisomer from the racemic mixture. We can resolve the problem by using enzymes for separation of racemic mixture. So these racemic mixtures are very tough to separate from using chemical method or chromatographic method. So enzymatic methods are highly specific to resolve the mixture of racemic compounds. So enzyme catalyzed synthesis route often reduces the eliminated the need of chemical harsh substances or high temperature conditions. So in pharmaceutical industries or in chemical industry, drug synthesis industry, they need to use harsh reaction condition for production of drug molecules like high temperature, high pressure for production process, and even they can use toxic metal catalyst for the catalytic activity or catalyst reaction catalysis. So instead of using this type of harsh reaction condition, you can use enzymes in a mild reaction conditions. Enzymes requires only aqueous buffer and a suitable pH and normal room temperature. So we can avoid the use of toxic chemicals and use of high pressure and temperature. We can reduce the cost of the product also. And for example, if you want to convert nitriles into a amide, so nitriles into amide, we need to break the C triple bond in C triple bond. It requires high acidic conditions or high temperature and pressure. So during that time, the formation of byproduct like HCN are more common. If you use in place of this harsh reaction conditions, use biocatalysis like nitrolysis and nitrile hydrolysis, we can easily carry out in mild reaction conditions. So without any formation of toxic byproduct or without using any high temperature reaction conditions. Another important thing in to achieve the use of this biotransformation method in the pharmaceutical industry, because we can approach in interdisciplinary manner. We can reduce the cost, economically benefit and advantages to implement biocatalysis in industry. For example, penicillin are synthesized semi-synthetically using the enzyme in a part of reaction, steps of reaction, and chemical steps in part of the reactions. Finally, they are synthesizing the penicillin. So like that, different antibiotics require synthesis of active antibiotic molecules, require some of the simple enzymatic reactions like deacylation, acylation, deamination, amination. So these type of reactions in combination with chemical synthesis, in combination with biocatalytic synthesis, by using interdisciplinary approach, we can modify, we can simplify the process. So then it is advantageous and economically benefit for the cost reduction. So some of the here we can discuss some of the important enzymes which are commonly used in pharmaceutical industries for synthesis of intermediate for the production of active pharmaceutical ingredients like alcohol dehydrogenase plays a major role for the synthesis of asymmetric production of hydrophobic alcohols. Yes, selective alcohol dehydrogenase or selective alcohol dehydrogenase plays a very important role for synthesis of alcohol, keto to alcohols. And nitrile plays a very important role in synthesis of non-selective carboxylic acid, glycolic acid, gabapentine intermediates, mandelic acid. For nitriles also used for conversion of 3-cyanopyridine to nicotinic acid. So nitrile hydratase have many applications in synthesis of acids, amides, corresponding amides, 3-cyanopyridine to nicotinamide, nicotinamide, and acrylonitrile to acrylamide, acrylic acids to acetonitrile to acetamide, benzonitrile to benzonide, and S-selective alpha-amyl, alpha-methyl, phenylalanine, stereoselective amides. So amidases also plays very important role in production of nicotinic acid from 3-cyanopyridine pyridine, and benzoic acid, acrylic acid lithostats, phenylacetic acid, 6-amino penicillinic acid. So these are the commonly used for production of intermediate and transaminases. These transaminases plays a very important role for the synthesis of many intermediates like sitagliptin. Sitagliptin is a anti-diabetic drug molecules synthesized from single steps in conversion by using transaminase. R amino butaramide, S amino butaramide, droxidopa. So these are the therapeutic molecules or drug molecules, precursor for the synthesis of drug molecules. So lipase also plays very important role. So lipase, usually people are thinking that lipases are only used in food and processing industry. No, lipase plays a very important role in pharmaceutical industry for the esterification, de-esterification, resolution of racemic methyl ester, resolution of 1-methyl, or tetralone synthesis. So lipases, lipozyme calpi. This calpi is isolated and from and genetically modified. Lipase enzyme is available in the market. That's called as calbi enzyme. So this calbi isolated from candida. So another important enzyme is enantiomeric reductases. The reductases also plays very important role for the synthesis of non-natural amino acids and alpha and beta amino acid analogs, beta nitroacrylate, S enantiomers, quiniquidines, anti-malarial drugs, and chiral starting material for the synthesis of different drug molecules. So using these enzymes, people already synthesizing antiviral molecules, anti-HIV molecules, anti-malarial molecules, and they are also synthesizing neurotransmitter drugs. So only thing is we need to select the proper enzyme for particular reactions. So these five, six enzymes playing, 7 enzymes playing very important role in, especially in the synthesis of active pharmaceutical ingredient, precursor for the final drug productions. So only in how we can achieve or how we can develop develop or use these enzymes, and where can what are the steps involved in selection of these enzymes for the particular reactions. So different approaches are there. First, we need to screen the chemical transformation. For example, transamination reaction we are doing means we are taking the keto to keto and amide as a amine donor, and then we are synthesizing the amine. So first, we need to see the reactions, what type of enzymes required and what types of donors, what type of acceptors are required. Then we can utilize type of enzymes required for the invention of new product molecules. Then go for the development of small scale and go for the development of large scale synthesis and biochemical process. You can optimize the substrate concentration, buffer concentration, pH can be different optimization condition, and then develop a process. But sometimes this process may not feasible or not meet the cost of the production. Then we can go for improving the strain engineering and fermentation technology development and optimization of the fermentation condition to get good product, to get large quantity of the recombinant protein or the natural protein. The success of screening or success of use of enzymes for the catalytic activity depends upon the screening method. You can isolate the organism from microbes, microorganisms from soil sample. The success starts from the selection, identification of root, identification of chemical reaction, then selection of proper microorganisms or proper clone for conversion of substrate into a product. So some of the why we are looking for the microorganisms, because the microorganisms have diverse enzymes. They are continuously exposing to the different environmental conditions, different toxic chemicals, the different enzymes make changes, and the pharmaceutical complex molecules are able to convert the the microorganisms, enzymes have evolution. They become evolved to evolved to convert toxic chemicals into a other form. So that's what we are looking for the microorganisms. You can isolate from the soil samples. So we have isolated different enzymes for the application of for the production of alcohol dehydrogenase, nitrile hydratase, amidase, transaminase, lipase, and reductases using soil samples. And we have also collected the samples from the literature. I listed the organisms which are used in the used for this purpose based on the literature survey, based on isolation. We have developed a library, organism library. So you want to screen particular chemical reaction from the library of the organism, we will take and screen a particular plate methods or assay methods, either plate-based methods or the liquid culture-based methods in 96 well state, or high throughput screening methods can also be used for screening of microorganisms. So from the soil sample and available organisms in the culture collection center, we, we created the library of different organisms for different enzyme productions. Alcohol dehydrogenase producing enzymes, smart short chain alcohol dehydrogenase producing enzymes, long chain alcohol dehydrogenase producing enzymes, and so middle chain alcohol dehydrogenase producing enzymes, nitrile producing enzyme, amidase producing, transaminases, like that. We have created a one library. So if the chemical people ask for the particular reactions, which enzyme is suitable and what is the reaction. So then we will see the reaction and what type of enzymes is working for that reaction. We will go and take that library of organisms. We are screening by plate method or by 96 well plated method or high throughput screen method. We will get a hit. The small hit can change to develop a recombinant protein that can be used as a biocatalysis for large scale production. So these simple steps also very important to get a novel protein, novel enzyme for a particular drug synthesis. So why we are using microorganisms in pharmaceutical industry or in industries for large scale production? Because microbial enzymes are more stable, cost effective than plant and animal enzymes. So we can produce the through fermentation techniques in cost effective manner with less time and less manpower and less space requirement because of their high stability. And the optimization process, the process optimization, modification of process can be easily done in laboratory with fermentation technology. And we can also culture in large quantities in a short period of time using fermentation technology and biochemical diversity and susceptibility to gene manipulation. So these microbial enzymes or microbial genomes are really very easy to manipulate, are very easy to clone and express in a same homologous and heterologous expression system. So industries looking for new microbial strain in order to produce different enzymes to fulfill the current enzyme requirement. Why? Because the modifications and adaptations of enzymes for the particular substance, because pharmaceutical molecules becoming complexes. So these complexes can be synthesized using only existing enzymes or trying with the new varieties of enzymes which are present in the microbial genomes. So we developed around 4,000 in-house library of collections of 4,000 strains. These 4,000 strains are isolated from soil sample. Some of the strains are more than 3,500 strains isolated from the soil sample, and some of the strains we have purchased from the ATCC culture collection, culture, NCRM culture collection center, and different culture collection centers like DSM, the Japanese culture collection center. So all these potential organisms, we have created a library. So in this potential library, we used these organisms for screening of different varieties of enzymes. And we have optimized the protein production from the cells. The process is then developing our own small scale parallel to bench scale to reactor from 1 liter to 5 liters, 10 liter in laboratory fermenter. Then same scale up we have set up for scaled into higher level like 1 kg 1,000 liters, 10,000 liters, and 20,000 liters. So in our library, we have developed the organism. We have created a library of organisms producing alcohol dehydrogenase, nitrulase, nitrile hydrogenase, amylase, transaminase, and hydrolase. So these organisms are isolated by using the specific substrates for production of particular enzymes. So to get a particular biocatalysis or to success in the biocatalytic process or biotransformation process, we need to follow some steps. For first, identification of target reaction. So once we identify the target reaction, then we can go for the classical screening of organisms, either high throughput screening or plate methods or 96-well plate methods. Then the substrate range kinetic data analysis, substrate inhibition concentration, substrate kinetic prediction, reaction conditions optimization, structural analysis. Then we can go directly if the setup is okay for the wild type of organisms, we can go for directly for the, if it is a method is viable or cost effective, you can use whole wild type organisms as a biocatalyst. Either you can use whole cell as a biocatalyst, or you can immobilize the whole cell into in a suitable matrix and use as a biocatalyst, or you can purify the protein from the native organism and you can use that enzyme as a biocatalyst. Either if the methods are not feasible, are not meeting the cost because the native proteins are produced in very less quantity in the organisms. We need to modify or we need to express in large quantity by using recombinant DNA technology or biotechnological process. For that reason, we need to express the gene, engineering the protein, and engineering the cell or metabolic engineering. Then we can use engineered strain or engineered enzyme for the biocatalytic production. Then, if the optimization is over, then we can go for the large scale production in a economically sustain a cost effective manner by applying by optimizing all the parameters. So for achieving the product process we need to use different steps. So for development of recombinant clone, we need to identify gene, we need to express, we need to engineer the protein, we need to engineer the whole organism. For identification of gene, we are tried with the different approaches. If the organism sequence is available in the database, we can use directly that genome sequence. We can see the protein sequence. We can construct the, we can amplify the gene by designing the primer from the wild type organisms, or we can use that amplified product for overexpression. But we are isolating the gene from the microbes. That may the sequence may not available in the database. The sequence information is not present in elsewhere. In that case, it is difficult to clone and overexpression. Development of clone and overexpression of particular gene is difficult. So for that purpose, we have used different approaches like construction of genomic library and screening of library clones for selection of proper clone having a desired gene. So another method is reverse engineering or reverse approach like purification of protein from the wild type, and from the protein sequence we need to design the degenerative primer or amplification of gene, and that gene product is expressing in particular vectors, host, or we can do the complete genome sequence in order to understand better understanding of arrangement of gene, what are the genes responsible for that expression of the protein molecules, what are the regulatory genes, what are the other components which plays important role for expression of the gene. So different approaches also be used for some of the novel transaminases, gene amplifications. So this is different strategies we use to amplify, we use to identify the gene. If it is a gene sequence is available, then go for directly. If you develop a gene expression, first we need to take the native gene or native enzyme or purify the enzyme, determination of enzyme structure, enzyme property, or once the determination of enzyme property or the data available in the research bank or gene bank or bioinformatic data, we can go for the designing of primer, and using that primer we can amplify and construct a recombinant vector. That recombinant vectors commonly used for in expression system. We use PBAG vectors, PET system vectors with different hosts like E. coli BL21B3, E. coli architect express, E. coli Rosetta Gummi, E. coli Rel. So different hosts we used for expression of gene into a useful protein product, protein antibody enzyme product. So for that reason, we have also considered some of the important steps. Either we identify the metagenomic process or bioprocessing from the microorganism, or genes we have taken from the external organism. Once we get the natural biocatalysis, we are going for structural identification, homology modeling, and different models for identification of the proteins. Once the protein information is getting, so where we need to engineer, where we need to modify to get a stable protein, a stable enzyme, or to increase its efficiency, or to increase its selectivity, or to increase its stability, we are going to engineering that protein into a recombinant protein for the useful product production, either site-directed mutagenesis, direct evaluation, or enzyme modification for immobilization techniques. And then we will go for large-scale production, heterologous expression, bioreactor level production. So during development of recombinant clone or engineering of protein also important. Why? Because the cost, it has to meet the product cost, has to meet in industry. They are concentrating product cost also. So and use of that recombinant engine in large scale production, that should be stable for multiple purpose, reusability purpose. So that's what we need to engineer the protein or enzymes, improvement of kinetic property. So we can increase the activity by changing its kinetic property by modifying or mutating the enzymes, elimination of allosteric regulation. Some of the gene expressions depends upon the allosteric regulation. The protein expression depends upon allosteric regulation. So that modifications we can also do by genetic engineering. Enhancement of substrate and reactant specificity can also be increased. For example, some of the stereoselective enzymes are more specific for particular stereoselective, R selective, or less specific for S selective. We can completely remove the S selectivity. We can achieve only R selective compounds extraction, R selective compound substrate specificity. That can also be changed by engineering, by using genetic engineering. And we can also increase the thermostability of the enzyme for we can use high temperature for conversion. We can also optimize the pH sensitive enzymes. We can also use different method for engineering. We can also engineer the enzyme, because most of the stereoselective and enantioselective molecules are intermediates for synthetic drug molecules, requires organic phase to phase, because the extraction process will be easy. So we can modify enzymes, may be stable, or enzymes it is suitable to keep the reaction in organic solvents. So that can also be modified by engineering method, or we can also increase the thermostability for long use. So the biocatalytic process involves either use of single, either recombinant enzymes or native enzymes, but the steps are different. Sometimes we can use single enzymes for conversion of substrate into a product, or sometimes we need to modify the, or we need to engineer the whole gene, engineer the gene, or metabolic engineering is required to get the product from different multiple steps conversion. Or sometimes we can use cascade of reactions. So simultaneously we can keep two enzyme expression for conversion of substrate A into B and B into C. So in one step, so only one single enzyme is required. In metabolic engineering, or the conversion of carbon source into a particular product in multiple steps, we need to engineer in whole genome, or we need to engineer in genome only. Or some of the catalytic steps requires two enzymatic steps. For example, conversion of A to B substrate, for that one enzyme is required. Conversion of B to C, the second enzyme is required. And sometimes this second enzyme also requires some prosthetic groups. So cofactors. This cofactor regeneration also shows the cost increases. They may also contribute the increase of the cost. So that can also be cloned simultaneously expressed to regenerate the NADH, for example, in dehydrogenase reactions. So glucose dehydrogenases, formal dehydrogenases, leucine dehydrogenases are commonly used for regeneration of NADH into NADP. So that is, we can reduce the cost of NADH. So using different approaches or different method for conversion of substrate into a product are required. So in single step conversion, we have isolated, so it's our research work, we have isolated Paracoccus denitrificanes, which is able to produce nitralase, nitrile hydratase, and amidase. These nitrals are convert nitriles into a amides and carboxyl amine and carboxyl acid by nitrile hydratase. One step. In the second step, two enzymes are required for conversion of nitrile into a acid and ammonia, nitrile hydratase and amidase. We have confirmed the growth of the organism, utilization of nitrile and increase in the pH of the media. If the nitrile hydratase and amidase, these two enzymes are present in the organism, we used nitrile as a sole source of carbon and nitrogen without any additional source of carbon and nitrogen. This nitrile acts as a sole source of carbon and nitrogen. So this acid uses as a carbon source and amine act as a nitrogen source. Excess of nitrile, excess of ammonia in the media increases the pH. We confirm the growth, we confirm the increasing in the pH of the media, and we also confirm the liberation of ammonia. Based on liberation of ammonia, we have developed rapid and screening method using phenol indicator as a phenol red indicator for detection of color change. So it's a synthetic media, N9A, mineral salt media, having nitrile as a sole source of carbon and nitrogen. If the nitrile degrading enzymes are present, nitrile converting enzymes are present, they convert nitriles into acid and ammonia. The excess ammonia contribute in the change in the pH of the media. The phenol red is red in neutral pH. When the pH increases to alkali, it changes pink color. So this simple indicator plate method, we can screen many different nitrile degrading organisms like nitriles to amides like 3-signopyridines to nicotinamide and nicotinic acid. So like that, different methods are also available based on screening, pH change, based on product formation, based on color development. So these simple technique can be used to screen hundreds of samples to get a single hit. From that single hit, we can use or we can make it as a recombinant product in large scale. So this type of simple experiment also plays very important role for screening. So this organism shows amidase. So these amidase have two types of activity. One is hydrolytic activity. Another one is acyl transferase activity in presence of hydroxyl amine hydrochloride and acetamide. The amidase converts hydroxyl amine into a acetohydroxamic acid. So this acetohydroxamic acid is a drug molecules, one step conversion of drug molecule lithostat. It can be used as a drug for treatment of urinary tract infection. So this drug can be synthesized a single step by using microorganisms enzymes. So we optimized the substrate required for conversion of product into substrate into a product. Here we are using two substrates because acylation requires ping-pong mechanism, acetamide and hydroxylamine for conversion of acetamide hydroxylamine into acetohydroxamic acid. We have optimized the time required for conversion, and we confirm the product based on the HPLC analysis, and we have also confirmed the product by NMR and mass analysis. So these simple engines, we can grow the organisms in waste effluent containing a nitrite, and same cells, same cell, same whole cells used as a catalyst for conversion of for synthesis of acetoatomic acid in presence of substrate. So same enzymes will be immobilized in PVA alginate blended beads. So these beads having enzyme, having whole cell shows the activity of conversion of acetamide into acetohydronic acid. So same beads we used for 8 cycle with 80% of its activity. So this shows that the organism whole cells are immobilized. The enzyme can be used for repeated cycle for synthesis of drug molecules. So this work I have done in my PhD with my interest, use of degrading microbial toxic chemical degrading organisms into a conversion of a useful product. So then I moved to industry to use this type of technology, biotransformation technology for the synthesis of variety of non-natural amino acids and synthesis of drug molecules or precursor for the synthesis of drug molecules. So one of the important molecule S-aminobutyramide production. S-aminobutyramide is a it's synthesized from L-homoalanine. L-homoalanine is a non-natural amino acid that can be key chiral intermediate for synthesis of several important drugs like conversion of S-aminobutyramide, which is a intermediate precursor for the synthesis of anti-epileptic drug uh, levetiracetam and dabigatran. It can also be converted S-aminobutanol to synthesize anti-tuberculosis compound called ethambutol. The optical purity of this drug is critical for therapeutic safety and effect. If you select R selective isomer, so enantiomeric property will be not, and this will not act as an anti-epileptic drug, and it will not show any anti-epileptic drug activity. And the excess of this R form ethanol can cause us blindness. So for synthesis of API molecules, especially for active pharmaceutical molecules, in the synthesis of especially chiral molecules, they look for EE 100%. Enantiomeric excess should be 100% without contamination of other isomers. For example, we need only S isomer, it should be 100% yes. If it is R is at 1% also, they won't accept, because these molecules may act in other way. They may be inactive and they may cause us different effect. So that's why, especially for the enantiomeric drug molecules, they need specific isomers. So this specific isomer synthesis can be achieved only through enzymatic method only. So enzymatic methods only. So that's what the 309 is the precursor molecules for conversion of 2-butyric acid. So for that conversion, we need 309 deaminase or 309 dehydratase enzyme. Then from the 309 to 2-ketobutyric to S-butyric acid is converted, or SABAM, it is also called as SABAM 2-amino S2-amino butyric acid by using glutamate dehydrogenase or transaminase. If you use glutamate dehydrogenase, we need to regenerate NADH. NADH is cofactor is required. So NADH cofactor regeneration, we use glucose dehydrogenase, or we can also use leucine dehydrogenase. So if you see the structure of this, here we used in the first reaction, we used single enzyme for conversion of substrate into a product, amides into a acid. So here we are using couple of enzymes, two enzymes, threonine deaminase. Along with the threonine deaminase, we are also using transaminase, or we are also using glutamate dehydrogenase. If you use glutamate dehydrogenase, using glucose dehydrogenase required. The cascade of enzyme construct were developed on this cascade of enzyme expressing host used as a catalyst, whole cell used as a catalyst for synthesis of S2-amino butyric acid. So this is another modification. We have also done genetic engineering, metabolic engineering. In E. coli system, the glucose usually converted into acetyl CoA and enters into a citric acid cycle. and it forms the oxaloacetate. From the oxaloacetic acid, the aspartate 309, and from the 309 to ketobutyric acid is formed. So this conversion of aspartate to 309 requires 5 different enzymes, and from the 309 to ketobutyric requires 309 aminase or 309 dehydratase. So these pathways exist in the bacteria, but the flux of this formation of 309 is not that much because this regulate all amino acid synthesis are regulated by allosteric regulation. So we need to mutate the enzymes which are allosterically regulating the 309 accumulation or ketobutyrate accumulation. So first we have mutated the gene which are responsible for allosteric regulation of 309. Then we cloned 5 different genes which are required for conversion of aspartate to 309. So 309 is accumulating. The accumulated 309 is directly converted into a ketobutyrate because along with that gene we have cloned 309 dehydratase. So entire cascade of genes will be used for expression in one system. So once the ketobutyrate is formed, this ketobutyrate can be converted into a homoamine, a non-natural amino acid by using amynation reaction. This is amynation reaction. The amynation reaction is carried out by using transaminases or dehydrogenases. So once the L-alanine is formed, this L-alanine acts as a precursor specific. It is a L-specific. This acts as a precursor for synthesis of S-aminobutramide for the synthesis of anti-epileptic drug, and S-aminobutanol at reduction reaction acts as a precursor molecule for synthesis of tubercular drug molecule. So this this biotransformation reactions, involvement of cascade of reaction, genetic metabolic engineering, overexpression, and use of interdisciplinary. Up to here, homogeneous synthesis, it is a enzymatic method. Then we use the classical chemical method for conversion of this to final product. So using interdisciplinary science, interdisciplinary approaches, we can achieve the production of target molecules. So different vectors we have used for expression purpose, PBAD vector, the PET system vector, along with the vector should contains different tag molecules. These tag molecules like histidine tag molecules or GST tag molecules that is useful for purification of recombinant proteins. So 3-amine dehydratase, we have cloned, and aminotransferase and transaminases, glucose dehydrogenase, and glutamate dehydrogenase enzymes we cloned in this project, and we use this clone combined cascade of enzymes used for production of anti-epileptic drug, homoalanine to ketobutyrate and ketobutyrate to uh, ketobutyrate, ketobutyrate to homoalanine to S-aminobutramide and S-aminobutramide. The chiral amines, apart from these, some of the chiral amines, especially the great importance in pharmaceutical and agricultural industries. Around 40% of the chiral drugs are amines, around 30% of the pesticides are chiral amine molecules. So these chiral amines plays very important role. The synthesis of our chiral amines are very difficult and very challenging process. If you use synthetic chemical method for synthesis of chiral R amines, you will get both R and S racemic. The separation of R and racemic amines is very difficult with 99.9 purity. The only we can achieve by enzymatic method is possible. We have achieved that by using transaminase enzymes. So the drug molecule R-amino 1-butanol is a key intermediate for synthesis of treatment of HIV drugs and HIV drugs, and it's synthesized in green efficient biocatalytic process using whole cell as a biocatalyst for conversion of R-aminobutanol into a R-aminobutanol using transaminase enzymes. So this transaminase enzymes exist, especially omega transaminases plays very important role to achieve chiral amines. So, uh, in the synthesis of chiral amines, many transaminases have S specificity or selective transaminases are very less, very exploited and very less steady, making the production of R amines is a challenging. So, what we did is first we synthesized the racemic amines. From the racemic amines, we removed unrequired amine into a keto acid and required amine used extracted. For example, we have isolated the transaminase that is specific for S amine. So initially we synthesized the racemic amine, in the racemic mixture, the S specific amides are removed by using transaminase. Its transaminase, the reaction is a reversible reaction. So if the excess quantity of S is present in reaction mixture, it converts S amine into a keto acid. So R will be the remain same. So this achieved 100% EE, enantiomeric excess of R former amine by using racemization using transaminase. This novel transaminase we isolated from bacteria from the soil sample only, and we used this whole cell as a biocatalyst for racemization or for conversion of mixture into a single or 3-amino butanol. So this single 3 R amino butanol acts as an anti-drug precursor molecule for synthesis of antiviral, anti-HIV drug molecules. So this we have succeeded, we have succeeded in this project. We have isolated this organism from the soil sample only. From the soil sample, we screened with 1 ml 96 well plate. Then we went for large experiment and small scale reactor, bioreactors and small scale fermenter, and we went for large scale fermenter. From 1 ml of reaction, we went for 20,000 liter fermenter production with both having transaminase activity. This transaminase is a novel because we are keeping this reaction in a organic solvents. The enzyme is more stable. The organism is constitutively expressing this transaminases. So from the soil sample only, we isolated, we used this enzyme for synthesis of rabo or amino butanol drug, drug intermediate molecules. So we can achieve, but we need to search for particular enzymes. We needed patience to get from the soil to get from small scale to large scale production. Really, we produced this engine, we produced these cells in 20,000 liters with 80 to 90 kg cell pellet. So this 80 to 90 kg cell pellet, we used a large scale reaction for bioconversion. So apart from this project, I am also involved in other projects like synthesis of droxidopa. The drug droxidopa is also known as L-DOPA or L3O dihydroxyphenylserine L3O-DOPAs. This L3O-34-dihydroxyphenylserine DOPA is a chiral unnatural beta-hydroxy amino acid used for the treatment of Parkinson's disease. This droxidopa production of prodrug for norepinephrine used to increase the concentration of these neurotransmitters, and this dopa, doxidopa also acts as an intermediate for synthesis of new variety of neurotransmitters. We developed the continuous bioconversion system using whole cells or using recombinant enzyme E. coli expressed in E. coli. So this 309 aldolase isolated from different strain, that isolated gene are amplified. This amplified gene is cloned and expressed in E. coli system. This E. coli system whole cell used as a biocatalyst. And also we developed recombinant E. coli expressed 309 hydrolase with histidine tag protein enzyme. So this tag and some modifications, genetic engineering we have done for the immobilization purpose, and we tried with the different immobilization synthetic resins. So we got one important epoxy resin. We bonded that recombinant 309 hydrolase to epoxy resin. So this resin we used for large scale production in continuous process, multiple cycles. Even we used it 10 times without losing its activity. So this plays a very key role, because same enzyme you are using repeated cycle 10 times with same enzyme with the same product yield. So that plays very important role, and we can also avoid the product contamination. So if you use immobilized beads or immobilized engine, there is no formation of product contaminations, and isolation or separation of the product also very easy. So we used this immobilized engine for conversion of 3,4-dioxybenzylaldehyde into a dopant in presence of amine donor lysine. So L309 aldolase recombinant enzyme we use for conversion of this state. So the challenges in getting this biocatalysis or use of biocatalysis for production of useful product having many many drawbacks, and we need to struggle for we need to face different challenges. The opportunities associated with enzyme catalysis implementation will not only the considerable profit and marketability, but it will further have consider integration of biomimetic approaches. So we are mimicking the natural reactions which is exist in the living system into a other environment. So it is not an easy task or easy approach to exact copy expression or to get the exact copy of approaches. So it is very difficult process, but we need to get a success in this field. All the components should be in one part, like efficiency, profitability, reusability, stability of the engine, reusability engines. Then only we can get a multiple, we can carry out a different reactions in different scales in production level. So not only the genetic engineering or development of clone or development of process. So it involvement of different interdisciplinary activities like process engineering. Once we have developed the process in R&D, we need to optimize the process in large scale production. So optimization of bioreactor design, optimization of process conditions like temperature, solvent, extraction process, or techno-economic analysis, and increasing in the life assessments or increasing in the stability also very important. And the immobilization technique also plays very important role in use of same engine for repeated purpose to meet the cost. So improved stability, reusability, functionality of the belt catalyst, reduction of the processing steps, and separation of the molecules after simultaneous stimulation from the optimum design, and the separation of the product from the immobilized beads or extraction of the product from the reaction mixture also plays very important role. Apart from this, the protein engineering also plays very important role to increase the biocatalytic activity, improve the enzyme substrate affinity, solvent tolerability, thermostability, isolated on the separation of enzymes after completion of the reaction with the stable activity. That also plays very important role. So with all these optimization of condition only, we can achieve the stable biocatalytic process, and we can use this stable biocatalytic process, or we can implement this stable biocatalytic process for large scale production in continuous and cost effective manner. So with this, I conclude use of enzymes in biocatalytic purpose. So not only the production of drug molecules, we can also use some of the enzymes as a therapeutic agent. So we focus on use of catalytic activity in a useful manner. So already the existing catalytic activity can be used for the production of useful product. So I have worked in development of different catalysis, not only from this. So majorly I worked on dehydrogenases, transaminases, nitrile hydratases, amidases, and reductases. So all these enzymes are very important, especially for the synthesis of stereoisomers and enantiomers. So if you have any clarifications, we can discuss with these topics.
Okay. Thank you, sir, for a wonderful lecture. Put it in the most common language so that most of the people can understand. So I thank you once again for this wonderful lecture. So we have a couple of questions popping out.
Yes.
Can we use droxidopa in case of treating Alzheimer's disease? Whether you have any idea about it?
No, sir, we have concentrated on synthesis of this
Okay,
heliocellular drug uh,
fine,
enzymatic
fine,
method. So
fine.
I cannot assure that we can use alternative drug for that.
Okay. One more question, sir. It is like, what motivated you to go towards this field?
Sir, frankly, I will tell you.
In other words, maybe the idea is like, what are the career prospects? How did you choose this field? Because of better career prospects or purely your passion?
So the motivation for use of these microbes in a useful manner, it came when I was in ninth standard,
Oh, great.
because we are using lactobacillus for preparation of curd.
Mm-hmm.
So, so that made me interesting to this field, because the microorganisms, we are not seeing the microorganisms with naked eyes, but
Mm-hmm.
these microorganisms is converting the milk into a curd product. So
Mm-hmm.
our milk into a useful product. So how
Mm-hmm.
it is possible? I thinking from ninth standard only, I decided to join as a science, to join science, and then I took the BSc CBZ. Then I choose the biochemistry interestingly to understand the life mechanism, or to understand the metabolic pathways in living organisms, especially microbes. So then I took the challenge, use of microorganisms for treatment of treatment of toxic chemicals to understand the metabolic pathway. So that's what we isolated the DMF degrading enzymes and elucidated the catabolic pathway. So this degradation of dimethyl formamide into amine and formate requires four enzymes with four different steps. So we have characterized all the metabolites, and we have identified the enzymes, and we have uh, done whole genome sequence to understand the enzymes which are responsible, how they distributed in the genome, and what are the enzymes and what are the regulatory sequences required for to understand the entire catabolic pathway of dimethyl formamide. So it's an interesting research, sir. For me, it's a very interesting research. So like that same way, I used the microbes as a useful product, or microbial metabolites or microbial enzymes which are involved in metabolic pathway that are useful for to get a to get a drug molecule synthesis or beneficiary molecule synthesis.
Right. Thank you, sir. Sir, how can we select particular enzyme for the production of a drug if there are two or more similar enzymes are available?
Yes, sir. So different enzymes also available. By exposing the microbes into a particular drug, then it is easy to select. For
Okay.
example, if you want to synthesize nicotinamide,
Hmm.
use particular nitrile as a substrate. So
Hmm.
expose that organism into a particular drug. If it is inducing the enzyme, it will convert nitrile into a amide. By exposing, by selective media, or by taking enrichment culture technique, we can isolate the particular enzymes. And if the enzymes are showing same activity, even we can also choose based on the product formation, based on the activity also.
Mm-hmm. Sir, use of microbial enzymes in compared to other industry, how it is cost effective in pharmaceutical industry?
So when compared to chemical method, the enzymatic method are cost effective only. So
Okay.
only thing, we need to engineer that enzymes using biotechnological approach, fermentation approach. We need to optimize the conditions, or we need to modify, engineer the enzymes So we can use same enzyme for repeated purpose, or we can express in high concentration, high quantity of the protein. Like normally it is expressing in wild type means we can increase 100 times or 10 times more than the production. So
Okay.
we can reduce the cost. And one thing,
Mhm.
some of the we can also eliminate the some of the synthetic steps which are present in chemical synthesis. We can avoid by using enzymes also. So we can
Okay.
reduce the cost, but to apply, we need we need more experts. We need more
Mhm.
knowledge to overcome the different problems. So like
Mhm.
fermentation optimization, recombinant clone development, immobilization technology. So then only we can achieve some.
Okay.
So it is
Sir,
not
in
all the drug molecules. So especially for the enantiomer separation is very difficult. Synthesis and separation of enantiomeric molecules using chemical method is very difficult. But
mm,
enzymatic selectivity method, it is very easy to produce single isomer.
mhm. Sir, since these microbial enzymes are very, very important in corporate sector, how they are preserved, whether they are preserved as a lyophilized crystals or cDNA library they make.
They will make a clone, and they will store it as a clone in a different host, one thing, and they will produce a produce, and they will use lyophilized enzymes.
Okay.
The clones are, they will produce in different varieties. They construct also, they saved in lyophilized powder also, and they are also stored in different host also.
Mm-hmm.
They preserved, they have a different methods and different techniques they use to preserve, and they can produce the recombinant enzyme with 100% purity lyophilized lyophilized product also. So like Codaxis, they are more expert in transaminases and alpha dehydrogenases. Even Novozymes are more expert in lipases,
Okay.
dehydrogenases.
Mm-hmm.
They are world leading market shareholders.
Mm-hmm. Sir, are there any enzyme strategies? 1 minute. Are there any biologically important enzymes that are now being used in industry for cancer therapy?
So directly they are using some of the enzymes for synthesis of drug molecules,
Mm-hmm.
like dehydrogenases,
Mm-hmm.
reductases. They are not directly synthesizing the drug molecules, they are synthesizing the intermediate molecules.
Okay.
So for sitagliptin, they are using for diabetic, anti-diabetic diabetic treatment.
Mm-hmm.
Sitagliptin, that synthesized from enzymatic only.
Okay.
The acrylamide, whatever we are using acrylamide, that is also from enzymatic method only.
Mm-hmm.
Higher sense. Higher sense. Fire.
Okay. I think that was a disturbance, it seems. Right. Sir, since I am not getting any more questions, so I'd like to take this opportunity to express my gratitude and extend a warm thank you for being with us. Even in your busy schedule, you spent your time and you shared your experience for more than 80 minutes. So thank you for giving us your precious time, sir. I hope the students are benefited. Sir, I have a request. If students are interested in this field and have some, want to have some guidance, so do I have your permission to give your email ID to contact?
Yeah, definitely, sir. You
Okay.
can. I will share my email ID.
Okay, sir. Okay. Thank you, sir. With that remark, on behalf of our Department of Biotechnology and Bioinformatics and organization committee of this international webinar series, I extend my warm gratitude and a big thank you from on behalf of our university also, sir. Thank you very much for being with us.
Thank you, sir. Thank you very much for giving opportunity. Thank you, Dr. Santosh sir, and organizing team and Department of Biotechnology. Thank you. Thank you, one and all.
Thank you, sir. So that was the talk by Dr. Santosh Kumar M. So indeed, a very wonderful talk. Um, so with this end, we are coming to the flag end of this international webinar series. So it's at the end, it is my duty to extend everybody a big thank you. So starting from our Honorable Vice Chancellor, Professor B.P. Veerabhadrappa, and our Honorable Registrar, Professor S.S. Patil, who within a short notice, when we went with the proposal, they gave a go ahead permission, and also they sanctioned us for the expenditure that will be incurred for this particular webinar. So, and then I really reached out to different people, like, and whoever I reached with a single phone call in a single attempt, they agreed to my request, and they spent their precious time and shared their precious knowledge with us. So starting from Dr. Kshitish Acharya, Dr. Dr. Darshan, Dr. Bharat, Dr. Prakash, Dr. Sharath, then Dr. Krishna Reddy, and Dr. Atik Khalid, Dr. Santosh. So all of them, they spent even in their busy schedule, even they are present in different time zone, it's very difficult for them to adjust to the schedule that what we put in Indian Standard Time, but still they wholeheartedly acknowledged my request, and then they extended their support in the form of their lecture, precious knowledge sharing. So a big thank you to all of those experts who came over here and conducted their scientific deliberations. So and then I cannot leave without thanking our chairman as well as convener of this international webinar, Dr. V. Krishna, who is the backbone, and he gave the very much needed support and encouragement to go ahead with this particular webinar. Then I extend my thanks to all my colleagues, Professor Riaz Mahmood, Professor Y.L. Ramchandra, my own colleague Dr. H.M. Kumarswamy, who was the other day, he was here. So I extend my heartfelt thanks to all of them for their moral support. And I especially want to mention Hemant Kelkar and Dr. Bharat also, who have also helped me in extending the technical tips, how to arrange, how to go for YouTube Live, how to configure the things, how to configure website links, everything. So with their guidance, I could able to coordinate multiple efforts. So I'd like to extend my thanks to one of my students, Deepthi Bharadwaj, because when I am in a Zoom session, I cannot see what is going on in there in YouTube Live. So she was there all the time upon my request, and she used to forward me all the questions and comments that are popping up in the YouTube channel. So I extend my heartfelt thanks to Deepthi. And then most important, once again, Dr. Pramod from Davangere University Biochemistry Department. So he's the one who gave the idea to have multiple front for communication. So, and also he helped me in configuring some of the things, establishing a WhatsApp group, then how to share the YouTube link, etc., etc. So a big thank you for him. So, and then I extend my heartfelt thanks to all those who have directly or indirectly helped me in arranging this particular webinar series. So, and last but not the least, the people from different parts of the world. So we have a registration from Singapore, Japan, and Brazil, and US. So in all this, from all these different part of the globe, people registered to us, there are, as of now, there are more than 325 registered candidates, and all our videos starting from the day one have garnered an average of 200 views in YouTube, and also an average of 50 participants in the Zoom session except me. So by that, you can say that out of 330, at least we have a view of 260 to 270 for all the session. So that gives an idea that how eagerly people are logging into, are tuning into my channel, and logging into the Zoom session and getting the benefit of this webinar. So I thank all the participants who make this webinar a grand success. And once again, from my side, a big thank you for all. Now, at the end of the seminar, I want to share something with you. Like, if you come to this particular site, sites.google.com, the address is there on your screen. So if you come to this site, I have created a link there for submitting your feedback. So if you click it there, it will take you to a section where, following the link there, you can go to the feedback forum, which is already published online. So you have to submit your feedback. So it will take us some 5 working days to send you the digitally signed certificates for all of you. So thanks for being with us. And with that remark, it's a goodbye from us. Thank you very much for being with us. So hope you people will support us like this in future also. We'll be coming up with one more webinar purely focused on phytochemistry and pharmacology. So please be with us and encourage us like this only. So with this remark, I beg adieu, and thank you for being with us. Thank you very much.